everybody, welcome to another edition of the Lab 207 webcast. My name is Mr. Kite. I'll be hanging out with you today as we start a new series. We're moving in. It's going to be a pretty short series, but hopefully it'll be a good one. We're going to be talking about viruses today. I'm going to tell you now that I got some stuff on a hot plate in the background, so if you hear some buzzing and bubbling, that's what's going on. So starting out, always got to get our objectives for the day. There are 3 of them. Here are the things I need you to know or be able to do by the end of this video. First of all, explain why viruses are not considered living. Second, describe the structure of a virus. And finally, compare and contrast viral reproductive cycles. There are 3 of them. So let's go ahead and get going. This might be a pretty quick video. I know I always say that. Sorry. But here we go. So first thing, viruses. You think of viruses as being these terribly infectious agents that go around killing millions of people worldwide, so they must be alive, right? Well, not so much. Some of the characteristics that all living things have is they need to be able to reproduce, they need to use energy, they need to respond to their environment. There are several qualities that living things must be able to do. Viruses do not do any of these things. Viruses are very simple molecules that consist of a nucleic acid and a protein. We'll talk about that in a second. But viruses don't use any sort of energy. They don't have to eat. They don't really move per se. They cannot reproduce on their own. They do not grow. So all of the characteristics of life, there are not any of them that viruses fulfill. Maybe there might be one that they fulfill. But in order to be alive, an organism has to exhibit all 5 characteristics of life. Viruses do not exhibit more than 1 or 2 of them. So just recognize that though they cause tremendous damage worldwide, viruses are not living organisms. Second thing, let's talk about the anatomy of our beast. I'm going to make a couple notes on my slide here just to show you what we're talking about. This is a structure of a bacteriophage. That's a specific type of virus that infects bacteria. So viruses are all going to look a little different from each other, but in general, all of them are going to have some sort of nucleic acid. Now, that could be double-stranded DNA, it could be single-stranded DNA, it could be double-stranded RNA, it could be single-stranded RNA. So there are a bunch of different variations of nucleic acid, but they all have nucleic acid in the middle, and they will all have some sort of protein coat around the outside. The protein coat is known as a capsid. Now, once you get beyond that, all viruses are a little different. A lot of viruses have a viral envelope, and what that is it's a membrane around the capsid, and this membrane usually comes from the host cell that they have infected. So you could have, always going to have nucleic acid, always going to have a capsid. That capsid could be covered by a membranous viral envelope, and then beyond that there's going to be all kinds of variations. Some might have some internal proteins right there. Some could have a neck with a tail sheath set up. This whole assembly right here helps our bacteriophage to attach to bacteria. So just recognize all viruses are going to have some sort of nucleic acid and a capsid, and then beyond that they start to get a lot of variation between them. Now, the thing that makes viruses so damaging are their reproductive cycles. There are 3 major reproductive cycles that you need to be aware of. So I'm going to go through each one of them, then after that we'll be finished up for the day. The first cycle you need to know is the Lytic cycle, and this is the simplest of all of the viral reproductive cycles. Let me just take you through the steps here. So all 3 of the viral reproductive cycles are going to start right here with attachment. Our virus is going to attach to the membrane of our host cell. Usually it's going to dock up and exploit some membrane receptors on the outside of the cell. It's going to confuse them and say, hey, look, I'm something that you would normally attach to, so they allow it to bind. Once it binds, it injects its DNA or RNA, whatever nucleic acid it has, it injects that into the host cell, and a couple things happen. The genetic material of the host cell could be broken down, but either way, the genetic material of the virus is now inside the host cell. After that has happened, that genetic material hijacks the machinery of the host cell. So this is a piece of RNA we talked about in previous videos. RNA gets translated into proteins. So what happens is the host cell starts reading this piece of viral RNA right there. As it reads it off, it starts to construct all of the pieces of our virus. If you look right here, you've got all the different parts of the virus being built in there. So we've got like a hijacking mechanism going on where that genetic code is universal. This code right here, even though it is viral DNA, our regular host cell can read it and use that information and start building the pieces of our virus. As the pieces of the virus get built, they're going to start to assemble, and once there are too many of them inside of the cell explodes. This is lysis right here. In a lot of diseases, well, quick example is herpes. With herpes, you get cold sores and things like that. The reason you get cold sores is because all of those cells on the skin are exploding and releasing viruses. Really gross, but true. And once those viruses are released, the cell is dead, and those viruses can go off to continue the cycle on. That is like quick and dirty viral infection right there. Now there is a sneakier way of going about things called the lysogenic cycle. Lysogenic cycle looks something like this. We got this little figure 8 going on here. Let me take you through the pieces of it. Right here is where your virus is going to attach. Now, once that virus is attached, one of two things can happen. One, we can go straight into the lytic cycle and start exploding cells, just like we talked about. Or, this is sneakier. The genetic material from the virus actually gets incorporated into the genetic material of the host cell. So it's not doing any damage, it's just hanging out. At this point, that little piece of DNA right there is called a provirus, and that is because we are not yet to a virus. Pro means before. So this viral DNA could hang out in the genome of our cell forever. I mean, once you get infected with a lot of viral diseases, you may not get sick again, but their DNA is still lingering inside of the cells that they have infected. So every time a cell divides, that provirus gets replicated and copied. And what will happen is this provirus, or this prophage, will hang out until some stressful event occurs. When that stressful event occurs, that virus right there, or the viral DNA, will pop out and start being copied and go into the lytic cycle. So cells can hang out in the lysogenic cycle and not be sick, not cause any damage for a long time, and then some stressful event will cause that viral DNA to pop out, start being copied, and we jump into the lytic cycle. So it's totally possible that somebody could be infected and have no idea that they're infected. It is also possible that they could be exhibiting the lytic and the lysogenic cycle all at the same time. Now, the most complicated reproductive cycle is that of a retrovirus. The best known retrovirus is HIV, human immunodeficiency virus, and it is called a retrovirus because it works backwards. Usually, RNA is injected into the cell, and that RNA just immediately gets copied or gets incorporated into the genome of the bacteria. With HIV, here's what you got. Our HIV contains several important things. Inside of it, you have got HIV RNA. You have also got an enzyme called reverse transcriptase, which I do not see on my diagram, but it's called reverse transcriptase. And what happens is when HIV binds and releases all of its pieces parts into the cell, it initially releases that RNA. Once that RNA is out, that enzyme reverse transcriptase reads that RNA backwards and makes a copy of DNA. So normally DNA is read to make RNA. In the case of HIV, RNA is read to make DNA. That DNA is then incorporated into the genome of the host cell through an enzyme called integrase. So integrase essentially cuts up the DNA of the host cell and sticks in our copy of HIV DNA. So every time that cell divides and replicates, the genes for that HIV are going to be replicated as well. Those genes will then hang out in the genome of the host cell, and as they're hanging out in there, just like normal transcription and translation the whole time, you have got that DNA being copied to make RNA and whatever proteins are needed for that HIV. You got the HIV proteins right here. Those HIV proteins and RNA are going to bud off, stealing some of the cell's membrane, and then go out to infect new cells. So this is basically HIV right here. As long as somebody is infected, but the body is able to produce cells as quickly as they're being destroyed by viruses being made, they're not going to show any signs. It's once the viral load becomes too big and cells start dying quicker than the body can reproduce them that somebody actually goes from HIV into AIDS. But we'll talk about that more later on. So just recognize that normally the body works DNA into RNA. HIV goes backwards. It puts in RNA. That RNA gets copied into DNA. The DNA gets stuck into the host DNA, is then copied into RNA and the associated proteins, which become part of the HIV virus that buds off and goes to infect new cells. I think that's pretty much it. So remember, viruses aren't living. Remember that essentially viruses are nucleic acid and a protein capsid. There are 3 cycles: the Lytic cycle, which pretty much kills a cell immediately by hijacking its machinery. You've got the Lysogenic cycle, where the genetic material of the virus gets incorporated into the host cell genetic material and hangs out until some stressful event sends it into the lytic cycle. And then you have got the reproductive cycle of a retrovirus, where we are working backwards. Hopefully you found this little tutorial helpful. Thanks for joining us. I'm Mr. Kite. This is the Lab 207 webcast. We'll see you again.